Hello, I'm Neil Clark, and in this talk I'm going to look at benchmarking in the field of bioinformatics. Now, there is a wealth of biological data that has flooded in since the development of high throughput technologies. This data has opened up a whole new line of biological questions that are now addressable. Now, in the field of bioinformatics, which is a burgeoning field, there are lots of work happening. There is multiple algorithms and tools designed to address each biological question. So then, given some data and a biological question and the wealth of available tools and algorithms, how do we go about deciding which algorithm or which tool to use? The aim of benchmarking in bioinformatics is to provide a quantitative, objective comparison between the algorithms and tools, which is designed to assess their suitability for a particular biological question. So benchmarking is a systematic and objective method for assessing the available tools and algorithms for a specific biological question. Typically, the way that benchmarking works is by the use of a gold standard. Now, the gold standard is a ground truth to which by which each of the algorithms and tools are compared. I thought we might start our look into benchmarking by examining benchmarking in a completely different field, in the field of computer science, where benchmarking is quite well developed. Computer systems are usually benchmarked by measuring the time required to complete specific computer programs. A good example of this is the Standard Performance Evaluation Corporation CPU 2006 suite. You can read more about that at the website indicated here. On the right, in a figure there, I'm showing a published result of one of the CPU 2006 suite benchmark tests of a computer system. So if we look on the bottom left here, we see a description of the hardware of the system, the CPU and the memory, etc. And to the right of that, there is a description of the software installed on the system. In this case, it's a Linux operating system. Then above, we have the results of the benchmark tests. And there are a number of these. For example, 401.bzip2 is a benchmark test that measures the speed of the system in compressing files. And what is plotted here is the time to complete the compression, a standard compression, as compared to a reference time. Down here, with the 458.sjeng benchmark, we're testing the artificial intelligence capabilities of the computer system. And this is done by running a standard chess program. And the time to complete the chess problem is measured and compared to a reference. Up here with 403.gcc, this benchmark test measures the time that the system takes to compile C code, for example. So you see here we have a number of different benchmark tests. Each one quantifies the capabilities of the computer system in various aspects of computing. And this is done in a very standardized way, so so that all computer systems, any computer system, can be compared in a consistent, standardized, objective manner. And its important aspect of this benchmark suite is that there are a number of different benchmarks designed to test different aspects of the computer system. And this kind of stands as an analogy for benchmarking in bioinformatics.